Hello everyone, I'm Yin Sun from Southeast University, China. Hope you all healthy. It's a pity that I was unable to attend the conference in Paris due to COVID-19, but thanks to the committee, I still have the opportunity to participate online. The title of my report is Application of Electrospun Nanofibers in Evaluating the Level of Oxidative Stress in Autistic Children. My report mainly includes 4 parts: background, methodology, application, and acknowledgement. First, let's look at autism spectrum disorder, shortly named ASD. ASD is a range of neurodevelopmental problems without certain causes. It's characterized by 2 symptom dimensions of impairments in social communication and behavioral patterns. ASD has a high incidence of 1 in 59, and there is a tendency to increase year by year. Unfortunately, there is no cure for ASD, but it can be improved by intervention. Here is a picture that ASD children and their parents, volunteers, are doing exercise to improve their symptoms. We think that we need some methods to evaluate the effect of improvement. In addition to the traditional ABC scale, CARS scale, etc., we hope to develop a biochemical evaluation method to expand the material basis of evaluation. Studies have shown that ASD is closely related to the concentrations of 8-OHDG and 8-OXOG in human body. 8-OHDG and 8-OXOG, oxidized by hydroxyl radical attack at the C8 sites of damaged DNA and RNA, are recognized as biomarkers of oxidative damage to DNA and RNA by endogenous and exogenous factors. Measuring the levels of 8-OHDG and 8-OXOG in people with autism is expected to be a way to assess the degree of autism. So, what kind of sample can be measured? The answer may be biological samples. Biological samples usually include 2 categories: invasive samples and non-invasive samples. Invasive samples mainly refer to blood, and non-invasive samples include saliva, urine, and feces. The collection and assay of non-invasive samples are friendly to humans, especially children with autism. Although all the 3 types of non-invasive samples are studied in our lab, feces are difficult for testers to accept because of the disgusting smell, and there are too many enzymes in saliva. Therefore, the most common non-invasive samples we test are urine. But the trace concentration and complex matrix makes the analysis very difficult. This page of PowerPoint shows an analytical scheme composing of 4 steps: sampling, pretreatment, measurement, and calculation. The time required for each step varies, and we can see from the PowerPoint. Sampling takes 6% of the whole time, pretreatment 61%, measurement 6%, and calculation 27%. It tells that the step of pretreatment takes most of the time, and it becomes the bottleneck of the whole process. Traditional pretreatment methods include liquid-liquid extraction and solid-phase extraction. Solid-phase extraction requires the use of commercial cartridges and nitrogen evaporators. Commercial cartridges are very expensive, and the nitrogen blowing process is extremely time-consuming. So what can we do? We have developed a packed fiber solid phase extraction, shortly named PFSPE, sample pretreatment method, that integrates separation, purification, and concentration into one step. The method largely cuts off the time. We prepare nanofibers by the method of electrospinning. Electrospun nanofibers have many advantages, including very large surface area to volume ratio, flexibility in surface functionalities, superior mechanical performance, and versatility of design. This is a schematic diagram of electrospinning. First, we pack the spinning solution in a syringe. Use the syringe's pump to push the solution to form droplets. Then, the droplets form Taylor coils under the action of the electric field force between the syringe nozzle and the collecting plate. The droplets are further stretched into streams. As the thin streams evaporate during the spraying process, they are collected and solidified on the collecting plate. We have prepared nanofibers of different morphologies, including porous nanofibers, aligned nanofibers, hollow nanofibers, and smooth nanofibers. Different polymers can be electrospun into different nanofibers. Polymers can be spun individually or mixed with other polymers or particles to obtain different extraction properties. If you are interested, you can check our papers for details. Considering the high polarity of 8-OHDG and 8-OXOG, polystyrene polypyrrole, shortly named PSPPY, electrospun nanofibers, was chosen here. The selective interactions between PSPPY nanofibers and target molecules was utilized to achieve efficient and rapid extraction of 8-OHDG and 8-OXOG. This page of PowerPoint is the schematic flowchart of PFSPE procedure of 8-OHDG and 8-OXOG. On the left of the page of PowerPoint is a picture of semi-automatic extraction device developed by our lab, and we can also tell from the picture that the procedure includes activation, loading, rinsing, elution, and equipment analysis steps. DPBA solution was introduced in the loading and rinsing steps to enhance the interactions between DPBA solution and target molecules. The next page shows the mechanism of the interactions. DPBA solution can form BOH interactions and BN coordination with 8-OHDG and 8-OXOG. And combined with LC mass, the determination of 8-OHDG and 8-OXOG in urine was realized. The method was successfully applied to detect 8-OHDG and 8-OXOG in urine of autistic children. A cohort study of ASD children and normal children was conducted. The experimental group was 23 ASD children aged 4 to 7, recruited from a rehabilitation institution in Suzhou, China. The control group was 23 age-matched normal children recruited from a kindergarten in Suzhou, China. Morning urine samples of the subjects were collected and detected according to the above targeted pretreatment and analysis methods. From the results, we found that the urinary concentrations of 8-OHDG and 8-OXOG of autistic children was significantly higher than those of normal children, indicating that a decrease in the protection against the hydroxyl radical may be a fundamental mechanism in ASD. Urinary 8-OHDG and 8-OXOG concentrations were significantly higher in children with severe autism than in children with non-severe autism, but there was no significant difference between children with mild and moderate autism. This suggests that abnormally high level of oxidative stress may predict severe autism, and the results was not shown in the PowerPoint. An ASD identification model was established by binary logistic regression, and the results shows that only 8-OHDG was in the equation. It may mean that 8-OHDG could be used as the identification index, and the accuracy and specificity were both over 90%. The AUC can be seen from the table. And this is the ROC curve of the two targets. The results provide empirical evidence for the oxidative damage mechanism theory of ASD and provide ideas for the development of new intervention methods for prognosis assessment. Our study was sponsored by the National Key Research and Development Plan and the National Science Foundation of China. Okay, I will end here. Thank you very much. Bye-bye.